Go back. We had skipped over some things in an adaptive that we need to make sure that we address. So, where is it? Related to antibodies. So, as we said, right, the very first time, there's going to be a time frame where those things that we talked about are going to happen, right? A dendritic cell is got to present to T cells. T cells may need to activate B cells as well before you get this antibodies actually being secreted, actually being detectable in the blood, right? So notice this lag period could be anywhere from 7 to 14 days or 10 to 12 like they have on here. This is when you've never seen the antigen before, right? So this is when a naive B cell has to be activated. Most commonly, they're going to produce the class IgM first, then often they're going to class switch to G. And that means they're changing that portion of the antibody, the B cell is. The second time we're exposed, who is starting off this response? Is it a naive B cell? No, it's who? Memory cells, right? Memory B cells, memory helper T cells are helping out if need be, right? And the response is that much faster. Really quick, lots of antibodies produced. This happens every time you're exposed to the same antigen. You're going to produce more and more antibodies. You're going to have a more and more aggressive response each time. So B cells, as we've talked about, right, they're going to differentiate into plasma cells. This electron micrograph shows those really big endoplasmic reticulum because antibodies are proteins, right, that we're secreting. This is what it looks at under a light microscope with the standard staining. You can't really tell one lymphocyte from another, right? This could be a B cell or a T cell or even a natural killer cell. Nice purple nucleus, little light blue cytoplasm. This is a scanning electron micrograph where you can see all the receptors and such on the surface. Looks kind of scary, huh? After a few days, they go through the process of apoptosis. Basically, their job is done, right? Because apoptosis means what? Cell death without what? Without inflammation, right? This is their time has ended. But we're going to continue to activate B cells. They're going to continue to proliferate into memory cells and plasma cells. We're going to continue to secrete antibodies, and that concentration, that titer, is going to continue to increase until we've eliminated the antigen, right? And then these cells are going to die off, except for our memory cells. We don't maintain the army. It's too expensive, right? Feeding them is the tough thing. So one of the things that happens is that as the B cells are proliferating, they tweak their DNA just a little bit, right? In an effort to make that antibody's binding site more able to hold on to the antigen even better, what we refer to as affinity. And this is a process of natural selection. If you're really good at binding, then you'll continue on. Your lineage will continue on. If you're not, you're going to be eliminated. So notice they have pluses to denote the level of binding, right? So this guy is pretty good. He's a 2+. Then some of his cells divide, right, and they mutate a little bit on purpose. And this guy ends up being less able to really hold on to that antigen, has less affinity. So notice his lineage is discontinued, right? He's not going to continue to multiply. This guy, who's still a 2+, proliferates, and some of his prodigy become 3+. Right? That's better than the 2+ now, right? These guys are the best. So this lineage would be continued, right? Be stimulated, right? You guys are making the best antibody. It's binding the best affinity, holding on to that antigen, right? This is the one we really want. This process is referred to as affinity maturation. right? So although we talk about clones being genetically exactly the same, they're not truly exactly the same. So the other thing that happens is what we refer to as class switching, right? We start out with M, and when we look at the chromosome, we look at the information on the heavy chain, first up is M, right? They're always going to produce M. That's why M's on the surface of the B cell. That's the class of antibody. And that's the first one it's going to produce and secrete. But very often, especially in our lymph nodes, it's usually going to switch to G. And it literally cuts out, deletes the section of its DNA. So now it can only make what's past M and D, which happens to be G. The more common switch with your mucus-associated lymphoid tissue is a switch to A, because remember, this is the one we secrete in that dimer form. And then, as we'll talk about later, some of us are predisposed to producing E that experience allergy, right? Not our favorite. So vaccination really exploits this ability of ours to remember, right? And to get the response to be faster and more aggressive. When you're immunized, typically when you're vaccinated, you are given a weakened form of some type of antigen. Your body's responding just like we talked about, right? Those steps. You're getting antigen presentation by the dendritic cells to T cells. You maybe even activate B cells and they're producing antibodies. But all of these guys, B cells, the two different types of T cells, you're going to produce memory cells, right? If they're activated, you're going to produce memory cells. And that's what vaccination is getting us to do, get activated and produce memory, right? So that the next time you're exposed, whether it's naturally, right, that response is that much faster, right? You don't have to find naive ones. You've already got memory cells on hand. Much rapid response. And in some cases, you may even still have antibodies around. That'll help protect you. So now I'm going to jump all the way down to the bottom of the lecture and play this animation, and then we'll talk about it. This is the last bit for antibodies. And I'm definitely going to check the volume first. My last class, I blasted them. And I see in here it was like way high.
Human cells do not have enough DNA to have separate genes for each antibody molecule. Instead, different segments of DNA can be mixed and matched to form different antibodies. Light chains are made from V and J segments in the variable region and a constant region segment, whereas heavy chains are made from V, D, and J segments in the variable region and one of 5 different constant region segments. As the lymphocyte divides during maturation, its genes are rearranged. DNA sequences are assembled by the random selection of V and J gene segments for the light chains and V, D, and J segments for the heavy chains. The constant region of the molecule is encoded by a single C or constant gene segment for light chains and 5 different constant regions for heavy chains. The rearranged DNA in the mature B cell is transcribed into messenger RNA and translated into the light chain of the antibody. DNA sequences for the variable regions of each heavy chain are assembled by the random selection of a V gene segment, a D gene segment, and a J gene segment. The genes for the constant regions of heavy H chains have Greek letters designating IgG, gamma, IgM, U, IgA, alpha, IgE, epsilon, and IgD, delta. The DNA in the mature B cell is transcribed and translated into the heavy chain protein. The light chains are then attached to the heavy chains to form the functional antibody. Because of the large number of ways in which the genes can be rearranged, many different antibodies can be produced. So each B cell, I like to think, plays the lottery, right?
It picks out when it's born in the bone marrow, and this is true for T cells too, right? How they make their receptors that are antigen specific is that they're literally cutting out, rearranging segments of their DNA, right? So once they pick their numbers, there's no going back, right? So you have the heavy side, the heavy chain side, and the light chain side. And of course, it's just a copy of that on the other side. So this is B cell, they're saying number 1. This is its unique pick, it's its lottery numbers. B cell number 2, a totally different B cell, pick totally different set of numbers, right? And combine them. Now, antibodies are proteins, remember, and this information is encoded in the DNA. So that, and we have 2 variable regions that come together to form this antigen binding site. So you have the combination of the heavy chain and the light chain. That's what we refer to as combinational, here I'll put it, combinatorial associations, right? So the combination of the 2 gives us diversity right there, heavy chain and light chain. Still with me? Right? And then what we call gene rearrangement is what we just talked about, was picking those different genes and combining them together. Now the thing is, the second thing is that they don't precisely cut these pieces, right? This is what we refer to as imprecise joining. So remember, this V segment that they're saying is number 3 is a whole bunch of nucleotides, A, G's, T's, and C's. And then your messenger RNA is produced, and that's read by the ribosome in groups of 3. Remember when we talked about mutation and how changing one base can, especially if you even eliminate a base, will shift the reading frame and mess that whole code up? Well, they purposely don't join these together precisely. So right there you end up with variation. So even if another B cell picked 3, 1, and 2, and 2, and J, like picked both of these, this very same set, they would still not be the same because they don't put the pieces together precisely. Still with me? Okay. One of these, I know there's got to be something out there, animation or something wise that shows this. I think I remember seeing one, but I'm going to try and search for one for you guys. So there's that 3 levels of diversity. And then remember when I talked about affinity maturation, even after they've picked this, they're going to tweak it even more once they become activated, right? Little base substitutions to try and get greater binding. So they purposely mutate their cells. So how many different antigens do you think are out there? Number-wise. 100? 50? 1,000? Million? Billions, right? Probably in the orders of billions, because the other thing is do antigens stay the same? No, right? Viruses are constantly changing, bacteria are constantly changing. So if we didn't change as well, right, if we had a set amount of DNA and we didn't manipulate it like we do, we'd be stuck with just what we are born with. And we would not be able to change over time, right? So this system of literally mixing up our DNA, right, and tweaking it and mutating it to get to produce different proteins is it's paramount to how this part of our immune system can keep up with all the change and all the different pathogens that are out there. So that, we have said this before, but remember, B cells, when they leave the bone marrow, they've done this. They've made their B cell receptor, which is antibody class IgM. It's been checked out. Hopefully they're not recognized self-antigens. We release them to go to the secondary lymphoid organs and lymphoid tissue. The thymus, on the other hand, is going to finish the education of the T cells. Okay? And the thymus atrophies after puberty, and there was a belief that maybe it became non-functional. There is scientific evidence that it does continue to work, but not quite to the level and volume as it does before we reach puberty. Because think about it, being a young organism before puberty, there's a lot of stuff you haven't been exposed to yet. So your thymus is very active. It makes sense that it's very large, right? But then as we age, it's less active, doesn't need to maintain the large size that it had before puberty. All right, B cells and T cells distinctly recognize specific antigens by these receptors. So that's the end of adaptive. We're going to go over next time disorders, and so you'll notice, and actually, I've got the objectives up for you guys in the PowerPoint. And for homework, but I think if you guys want, we'll bang it out in class. I want you to, before class, Wednesday, so let's see. I guess I didn't post the next one for you guys. What day is it? I've lost track of days. You know what? They're down here. That's what happened. I hadn't moved them up yet. So Friday we did our thing. I gave you guys your points. Today we talked about antibody diversity and reviewed from before. There's our disorders. And in fact, I don't think I've added your guys' homework yet. So we finish up adaptive. So do your connect adaptives. Add those in. And then I'm also going to add links here, but they just go to our animations and immunology. There's 3 of them I want you to watch before class. But we're going to watch them right now, but anyone who's absent, so they know. And when you, as we go through these, especially when you watch this first one, you know this process that we've been talking about, presenting antigen, right? Activating T cells and then activating B cells and producing antibody. Think about that, especially as we watch this first one. So I'm just going to tag this first one and then I'll tag the rest after, if I can find it. Where's all our animations? There they are. So IgE mediated hypersensitivity. Before next class. So we have one chapter left, but it's shorter than the rest, and it's kind of review. And it'll make sense as we watch this first one. So I'm going to go ahead and stop this